So good afternoon, everyone. Let us continue our discussion on sustainable energy and life cycle analysis. Let us start the second week lecture notes. Let us revise that, and then we will go forward with the problem solving. So the sixth lecture is based on RISK assessment and LCA frameworks. So let us start. So sustainability sorry sustainability issues are basically complex, and understanding their interactions with equally complex engineered systems is best done through well structured analysis frameworks, and the risk based frameworks have traditionally been used in the characterization and prioritization of environmental issues. However, increasingly, life cycle frameworks are gaining prominence as a means of characterizing and understanding sustainability because of its inherent benefits. So, we will find those benefits in the coming classes. So, before proceeding, basically, we need to understand what is risk. So, understanding risk frameworks for environmental and sustainability issues requires an understanding of the language of risk, the tools used to quantify risk, and the use of risk in regulation. So, basically, risk is defined as the potential for an individual to suffer an adverse effect from an event. The concept of risk is used in many disciplines such as finance, engineering, health, and the precise definition of risk can vary depending on the application. So, risk is risk is potential for an individual to suffer an adverse, that is unfavorable effect from an event, and for a precise definition it will have separate understanding for various disciplines like finance, engineering, health, etc. So when we talk about environmental risks, basically, so the environmental risks can be grouped into primarily can be grouped into classified into three general categories. So one is risk involving voluntary exposure, one is risk associated with natural disaster, and one is risk involving involuntary exposure. So what is risk involving voluntary exposure? That is activities done for a living or for enjoyment, such as firefighting, skydiving, mountain climbing, bungee cord jumping, etc. So here the risk or danger is usually obvious, and the activity is usually done by free will. So we are actually voluntarily participating in the risk or the activity which has risk. So that's why, and also the risk is obvious. So we know what can happen, what can go wrong. So that is risk involving voluntary exposure. In the case of risk associated with natural disasters, so they are basically floods, hurricanes, earthquakes, meteorite hits, and other disasters which are beyond the human control. And the exposure to the effects of certain natural disasters can be exacerbated or intensified by actions such as living on a known earthquake fault or the slope of a volcano. So the probability of this disasters will increase if we are at a certain location which has a intensified output or occurrence of that disaster. And the third risk, basically it involves involuntary exposure. So that is basically where we cannot directly control the exposure. So it is an individual or entity which releases a compound into the environment, such as pesticides, which are known carcinogens or cancer causing substances. So they can potentially harm workers or members of the public who cannot directly control the exposure. So that is why it is involuntary. So basically there are three types. One is voluntary, one is involuntary, and one is associated with natural disasters. So these are the three types of environmental risk. So the release of chemicals to the environment, so they are basically can be of three types. One is released to the atmosphere, one is released to the surface water, and one is released to the land. So it is quite trivial, it is very obvious. And so we can say that environmental risk is basically a function of two things. One is hazard and one is exposure. So basically hazard is basically its potential or the vulnerability of something adverse happening, and exposure is the duration basically. So it is a function of both. So risk assessment, so risk is basically the likelihood of harm, including both acute as well as chronic effects. So acute is basically short term and chronic is long term effects to human health and the quality of human life, ecological systems, economic systems, etc. The risk assessment, so basically this is the process by which the form, dimension, and characteristics of risks are estimated. So this is basically assessing the risk, and it will utilize both qualitative as well as quantitative approaches. So we will find out later how it is calculated and assessed. And third is risk management, so the process by which a risk is controlled or reduced. So first we will assess and then we will try to manage. So Risk basically on basis of regulatory decisions. So it can have various definitions. So one is cancer endpoint. So that is basically when we consider 1 in 10 to the power minus 6. So this is a threshold value. So if it is more than that, then we are considering it as a risk. If it is less than that, so if it is minus 7 minus 8, so we are not considering it as a risk. So second is epidemiological basis for human risk calculation. So that is also a different way of how we calculate risk. So epidemiology is basically the study of the incidence, control, and growth of diseases in human beings. So for that, we have separate human risk calculation, and one is animal testing where human data are unavailable. So in most of the cases, when human data is not available, we go for animal testing. So here most data are animal. If we see, mostly the data are animal tested data, except for those rare situations where a human exposure event has been documented. So we basically try firstly on animal, and then we convert that and try to replicate the impact on a person or a human being basically. So let us see what is a familiar risk. So the reference point is basically 1 in a million into that is 1 into 10 to the power minus 6. So this is as per the USDA, that is United States Department of Agriculture. So according to them, uh, for a food additive to be a concern, it has to be greater than 1 into 10 to the power minus 6. So below it, it does not consider risk from a food additive to be a concern. So minimal risk is when it is between 10 to the power minus 10 to 10 to the power minus 12, and very high risk when it is 1 to 10 to the power minus 2. So 10 to the power 0 is 1. So 1, that is every event has an occurrence of risk. So that is 1 to 10 to the power minus 2. That is 1 in 100. Okay. So that is very high risk. And there are some of examples of various incidents which has a different types of risk, various events basically. So if we say, talk about drowning in a tub per year, so the risk is around 10 to the power minus 5 to 10 to the power minus 6. If we talk about death in airplane crash, it is between 10 to the power minus 5 to 10 to the power minus 6. Similar. When we talk about death by automobile accident, it is between it is in the it has a certain value 1.7 into 10 to the power minus 4. In death by lightning strike, it is 10 to the power minus 3 to 10 to the power minus 4. Death from smoking, 1 pack per day, it is 3.6 into 10 to the power minus 3. Cancer from 1 liter beer per day for 1 year, it is 5 into 10 to the power minus 5. Cancer from 1 peanut butter sandwich per day for 1 year, it is 1 into 10 to the power minus 5. So it is a good risk basically, greater than 10 to the power minus 6. Cancer from charbroiled steak per week for 1 year, 7 into 10 to the power minus 6. So it is also a, as per USDA, risk. Cancer from drinking water with 80 microgram per liter CHCl3, that is chloroform, per day, it is 6 into 10 to the power minus 7. So as per USDA, it is not considered a risk. So the basic principles basically for risk assessment, it is a determination of the probability that an adverse effect or unfavorable effect will be produced. All human activities have some sorts of risk, some risk, concerned mostly with risk from exposure to chemicals. Risk assessment, so the extended definition, it is a methodological approach where chemical exposures are identified, analyzed for dose response relationships, and quantified for risk estimates. So the ideal approach for a risk assessor is firstly calculate amount of chemical absorbed into human body. Secondly, compare the absorbed dose with EPA, that is Environmental Protection Agency's safe dose for non-carcinogens or not non-cancer causing substances, that is the reference dose. So EPA does not recognize a safe dose for carcinogens. Any level of exposure has some cancer risk basically. A risk specific dose is used by EPA to assess exposure to carcinogens. So there are separate methodologies for carcinogenic and non-carcinogenic substances. This is basically the dose response curve. So here if we see, there are three ranges in this graph. One is when there is no effect basically, so it is no effect range, and thereafter there is a threshold value of dose after which it it increase it increase and starts firstly it increase at a exponential rate, then it attains a linear rate, and then again it starts decreasing the rate of ascent, and finally it reaches saturation. So the region between the increase between the threshold and the saturation, it is basically range of increasing effect with increasing dose, and after it has reached saturation, so it is basically called the maximum effect range. So that is basically the dose response curve. So uh, so it is a very trivial curve, but it has a lot of meaning basically. So the cancer risks in the history of epidemiology, only a few environmental agents have been repeatedly and strongly linked to human cancer. So these are some of the substances which have been found to cause human cancer risks. So one is cigarette smoke, one is alcohol, one is ionizing radiation, one is some various types of drugs, occupational carcinogens such as asbestos, solvents, benzene, and also viruses such as hepatitis B, human T cell leukemia virus, human papilloma virus, etc. So the cancer risks suspected based on observational epidemiology. So here are some of the values. So many epidemiologists require enough increased risk by factor of 3. That is risk ratio of 3. So risk ratio is basically risk in exposed group divided by the risk in unexposed group. So if it crosses 3, so then only many epidemiologists take new cancer risks seriously. So here are some of the examples of events with the risk ratio. So high cholesterol diet, so that basically is occurs in male. So it has a risk ratio of 1.65, and it occurs at the colon. The site is colon. High fat diet in female, it has a risk ratio of 2, and it occurs at breast. Vasectomy, so it occurs in male, has a risk ratio of 1.6, and it occurs on the prostate. The tap water with chlorine having a value of 3.3 liter per day. So it occurs in both the genders, male and female, and has a risk ratio of 2 to 4. And the site is occurrence of the cancer, the site is bladder. The psychological stress work, that is also found in both the genders. So that is having a risk ratio of 5.5. So this is a very uh, serious event. So these are some of the events which require epidemiologists to study and find out solutions. So that basically occurs in colon rectal. Similarly is the case with high saturated fat diet in females, which is having a risk ratio of 6, which is maximum in these events, and it occurs in lung or lung in case of non-smoker. And if we talk about red meat, if we are having it 5 times per week, so the risk ratio is 2.5, and it is for both the gender, and it also occurs in colon. So these are some of the cancer risk suspected based on observational epidemiology. So so if we talk about risk, there are various types of definitions which need to be defined. So one is the applied dose. So it is the amount of chemical in a medium that is available for uptake. So basically the total amount of chemical in a medium available for uptake is the applied dose, and the percent, the fraction of that applied dose which is absorbed into the body. Okay? The amount of chemical which is a fraction of that applied dose absorbed by the body, it is the absorbed dose. And for that we need to consider route of exposure also, such as ingestion, that is through mouth or nose, skin, inhalation. Ingestion is basically through mouth, skin, and inhalation is through the nose. And then there is reference dose. So RFD, it is its acronym is RFD, and it is basically the dose of a non-carcinogen that is believed safe for humans, usually extrapolated from animal toxicity data. So these are some of the definitions. We will find out how they are important. So apart from that, there are various other definitions such as risk specific dose. So that is RSD. So that is the dose at which 1% in a million exposed people will develop cancer. That is risk equal to 1 by 10 to the power 6. So 10 to the power 6 is basically the number. So above that, it is considered a risk. Below that, it is not considered a risk for this cancer and all and food related issues. So reference dose is basically for non-carcinogen, that is non-cancer, and that is a dose of non-carcinogen that is believed safe. And the risk-specific dose is basically the dose at which 1% in million will develop cancer. So that is the difference. And hazard index HI, so that is the ratio of absorbed dose to the reference dose. So this is the ratio of this absorbed dose by reference dose. And if the value is less than equal to 1, it is an acceptable situation. Whereas if it is greater than 1, it needs the, so the chemical concentration needs to be decreased so that reaches below or equal to 1. And risk management, basically, we have one more definition. It is the process of factoring the risk assessment versus possible alternative actions. So this is the process of factoring the assessment versus alternative possible alternative actions. So that is basically what is risk management, and that can be done based on cost benefit considerations, consumer needs, alternative chemicals, and to determine how best to regulate exposure to the chemical. So these are some of the ways of risk management. So let us see. So risk management. So process at site with drinking water. So if we take the case of risk assessment process at the site with drinking water. So the first process will be the hazard identification. That is what the chemicals, what type of chemicals are present, concentrations, carcinogens or non-carcinogens. So this the hazard identification is the first step. The second step basically is the dose response assessment. So that is the dose response relationship, the data validation, and also discovery of RFD, that is reference dose, and also RSD, risk specific dose data from animal studies. That is the second step. The third step basically is the human exposure assessment. So here it needs to make exposure assumptions also, because we have to convert sometimes we have to convert the data from animal to directly to human. So we need some factors, some diluting factor or error factors has to be multiplied. So that is sometimes that is the case. And example for drinking water, say for example, let us take an example for drinking water exposure. So let us assume that there is 0.5 milligram per liter zinc in the drinking water, and the assumed adult water intake is 2 liter per day, and the adult weight is basically 70 kg. So how to calculate dose? So dose is basically we for finding dose basically we'll multiply this and this and divide it by this. So per kg, what is the milligram per day value? That is basically the dose. So if we multiply 0.5 into 2 and divide by 70, we will be getting 0.0143 milligram per kg per day. So that is basically the dose. So this milligram basically, so that is the milligram of zinc, and this kg, that is the unit weight of the adult person. Okay, so don't get confused. So this kg is coming from the body weight, and this milligram is coming from the substance, the harmful substance basically. So the zinc or any other chemical which is causing the risk. So that is the difference, and it is per day. So that is how we calculate dose. And the fourth step basically is the risk estimation. So there we go for the HI or hazard index. And also if the value is greater than 1, as we just discussed, we have to reduce it. We have to take if you see here, if it is greater than 1, then the chemical concentration needs to be decreased, okay? Or some action has to be done. So if we take here, see in this case, so basically we compare absorbed dose to reference dose or RSD, risk specific dose, and cleanup required if risk is greater than equal to 1 by 10 to the power 6 or hazard index is greater than 1. So cleanup is basically required, or reducing the chemical concentration. So for zinc example, let us take the reference dose as zero point 0.2 milligram per kg per day. Okay, so this is the reference dose, and the actual dose was basically absorbed dose. Basically it was 0.0143. This was basically the absorbed dose, 0.0143 milligram per kg per day. So if we divide this by the reference dose, that is 0.2 milligram per kg per day, we are getting a value of 0.0715. So this is definitely obviously less than equal to 1. So therefore, since a hazard index is less than equal to 1, so no cleanup is required in this case, and we assume no other significant contaminants are present. So the risk is acceptable. Okay, so we don't have to do any cleanup in this case. And so apart from that, so let us see the development of RFD, that is reference doses for safe human dosage. So for that case, we have to search animal toxicity data for NOAEL and LOAEL levels. So NOAEL basically refers to no observed adverse effect level, and LOAEL is basically the lowest observed adverse effect level. So these are some of the levels which we will find in the dose and the risk and dose relationship. Basically we will find it. So we will divide that, that is basically for the animal toxicity data. So we divide this NOAEL and LOAEL by uncertainty factors, that is which ranges between a number between 10 cube and 10 to the power 4, so that we can convert this animal toxicity to human toxicity, which is certain uncertainty factor. So these uncertainty factors basically are multiples or divisions of 10. So 110 is for data uncertainty, that is high to low dose extrapolation. The other 10 can be for interspecies extrapolation. So one more 10 can be for the extrapolation between routes of exposure, and one more 10 can be for the protection of sensitive individuals. So it is basically ranging from 10 cube to 10 to the power 4. So for example, if a dose had no adverse effect on a mouse at a 0.5 milligram per kg per day, then to find the safe human dose, so we will divide it by greater than equal to 10 cube. Okay, so that is the definition. So for getting the same safe human dose, we will divide it by greater than equal to 10 cube. So if we divide it by 10 cube, basically we will get 0.0005 milligram per kg per day. So that can be with some certainty can be said to be the no adverse effect, the dose having no adverse effect for human, or the safe human dose. Okay, so that's where the uncertainty factors help. And for determining MCLG, or maximum contaminant level goal for drinking water, so there are basically three categories. One category is known carcinogen or probable carcinogen. So here the maximum contaminant level goal, or MCLG, is 0. In the case of second category, the MCLG, or the maximum contaminant level goal, is calculated by dividing the DWEL, or drinking water equivalent level, by SF, that is slope factor. And in the third category, when there is no evidence of carcinogenicity, so there the MCLG is equal to the DWEL, or drinking water equivalent level. So the maximum contaminant level goal is mainly dependent on the carcinogen. This slope factor is also dependent on basically the carcinogen only. So when we are sure that there is carcinogen present, it is 0. When there is uncertainty, we use some slope factor. And when there is certainty that no evidence of carcinogenicity is there, so the maximum contaminant level goal, or MCLG, is directly equal to the drinking water equivalent level, or DWEL. So how to calculate the DWEL and MCLG basically? So establish the NOAEL or LOAEL from literature. So this we have to find the values from literature. Then we have to calculate the RFD, or reference dose, which is equal to NOAEL by SF. So NOAEL is no observed adverse effect level divided by the slope factor. So we'll get the reference dose in milligram per kg per day. And the DWEL basically, which is the uh, drinking water equivalent level, so it assumes no carcinogenicity and exposure medium in water, and the exposure medium is water. So that is basically reference dose, that is body weight in kg, divided by liter of water ingested per day. So that is how we calculate the DWEL level. And MCLG, as we just discussed, it is 0 for category 1 contaminants. For category 2 and 3, it assumes air and food intake besides water also. So that is basically RSC, or relative source contribution. And for if that is there, so we will basically calculate the MCLGS. For basis A, it is DWEL minus food contribution minus air contribution. So if only food and air is there, so we will use basis A. And if only water is there, we will use basis B. So basis A is MCLG equal to DWEL minus food contribution minus air contribution, and basis B is MCLG equal to DWEL into percent drinking water contribution. So let us start with the seventh lecture, which is the RISK assessment toxicology. So what is toxicology basically? It is the study or science of poisons. Okay, so toxicology is nothing but the study or the science of studying various material related to poisons. Okay? So the study of poisons is toxicology. So poisons can be chemicals that produce adverse or unfavorable effects such as death, and the adverse effects decrease the fitness of an organism. So that is the one of the characteristics of toxicology. Okay, so it adversely affects or decreases the fitness of an organism. Uh, so that is basically the ability to survive and reproduce. That's why it's a poison. And toxicology is the study of how chemicals reduce an organism's ability to survive and reproduce. So this is a standard definition. So in toxicology, there are various terms associated. So let us see their definitions one by one. So one is toxicant, one is toxin, one is acute, chronic, latency. So these are common in other topics also, but this toxicant and toxin is the one of the main definitions we have to understand in toxicology. So toxicant is basically a toxic substance that is man-made or results from human or anthropogenic related activities. So any human related anthropogenic activities, so from that if a toxic substance is produced, okay, so that is a toxicant. Say for example, if we have if we have prepared DDT, which is sprayed as insecticides or pesticides, or in the field, or any say for example coronavirus. So that was also, it is said that it was prepared in a lab. So that is basically a toxicant. So what is toxin? So toxin is a toxic substance produced by a living organism. So okay, so that is, in this case there is no contribution of human activity or anthropogenic activity. So toxin is basically a living organism created toxic substance, such as such as a snake poison basically. So there no man-made contribution is there. So that is a toxin. And toxicant is something which is related to human related activity related substance, toxic substance. So that is the difference. And then acute is basically, as we discussed, it is a short term, okay, so single exposure or response. And chronic is long term, okay, so that is continuous or repeated exposure. And latency basically, this is the time between exposure and response. So between exposure and development of symptoms and various uh, responses based on the exposure, time taken between the exposure and responses, it is basically the latency. So as per very famous philosophers such as Paracelsus, so everything is toxic. Okay, so there is nothing which is non-toxic substance. Everything is toxic. So according to them, according to him, basically all substances are poisons, but there is none which is not a poison. The right dose differentiates a poison from a remedy. Okay, so according to him, everything which is present in the environment or in Mother Earth, so all substances are poisons. Okay, but the right dose differentiates a poison from a remedy. So if the concentration is in the acceptable limit, so then it is a remedy. Okay, but if it is exceeding that, then if it is not in a right dose, so it is a poison. So that was as per the understanding of Paracelsus. And examples can be DDT, that is dichlorodiphenyl trichloroethane, lead, oxygen, water, everything. So in a certain concentration, safe concentration, right dose, it is okay. But if you are increasing that anywhere in the body, so that is not, or any other place, so that is not a good this one good thing. So it has a tendency of becoming a poison. So there are some of the characteristics of toxicants basically. So toxicant, as we just studied, it is a substance which is man-made or anthropogenic activity related produced. So it has various characteristics. So mainly 5 characteristics. So one is it must be in the environment. Secondly, it must be bioavailable and bioaccumulated. Thirdly, it must interact at the molecular site. Fourthly, it must cause metabolic dysfunction. And fifthly, must decrease the fitness of an individual organism. So these are some of the characteristics of toxicant. One, it must be in the environment. One must be bioavailable and bioaccumulated. Thirdly, must interact at a molecular site. Fourthly, must cause metabolic dysfunction. And fifthly, must decrease the fitness of an individual organism. So the release of toxic material, so that can both be uh, intentional as well as legal releases. So benefits can be agricultural, health, industrial waste. So as we studied DDT and lead, etc. So these are some of there may be some benefits such as agriculture, health, industrial waste, etc. But it has adverse effects also. So unfavorable effects are mainly the toxic responses when the dose is not higher than the right dose. Okay. So the environmental fate can be various types such as partition. So it can uh, separate entities. It can transport from one place to another, and it can also transform to something very obnoxious or poisonous. Okay, so that is the problem of toxic materials. So, so these are the environmental fates. So the routes of exposure can vary in human body. So one can be gastrointestinal or GI tract or dietary, food and water. One can be skin or dermal, that is outside. One can be from outside. One can be pulmonary, that is through lungs or respiratory atmosphere. One can be through the injection or experimental, that is accidental or intentional. So these are some of the routes of exposure, and its characteristics are like it basically this toxic substances, they acquire a large surface area and they're highly vascular, that is they impact the functioning of blood vessels. So that is the way how it can harm the human body. So the major systems for elimination of these toxicants basically or toxins. So they are basically renal or urine, through renal or urine, through biliary or liver, through pulmonary or lungs, and also through some other minor routes of elimination. So these are some of the ways through which these toxicants or toxins can be eliminated basically. And this how like they bind with various how they cause problems. So one is to binding, so they interfere with normal receptors, interference with normal receptor, such as ligand interactions, which are neuroreceptors, endocrine, these are basically glands which secrete hormones in the blood, then enzymes, transport, etc. So one can be through this, and second can be through interference with membrane functions such as neuromembranes, cell membranes, organelle membranes, etc. One can be through the interference with cellular energy production, that is through oxygen transport, oxidative phosphorylation, etc. One can be through binding to biomolecules such as enzymes, lipid peroxidation, depletion of ZSH, which is glutathione, and DNA, that is deoxyribonucleic acid. So the types of responses through for these toxic substances, this is toxicants and toxins. So that they firstly, obviously it can be death. Okay, so person can, it can be fatal, person will die. Second is organ system or tissue damage. Third is other types of diseases. Fourth is and various types of mishappenings occurring. So these are some of the responses, and the repair can be through functional cell replacement or through fibrosis, which is scarring lining or connective tissue. Okay, so the chemical toxicity, so this is toxicity to humans, and they can both be cancer or non-cancer impacts. And there can be toxicity to ecosystems, so that is impact on individual organisms, population level impacts, and also community level impacts. So many chemicals have been found to be human carcinogens, basically, such as arsenic, which causes lung or skin cancer. One is benzene, which results in blood or bone marrow cancer. One is hexavalent chromium, which results in again lung cancer. One is vinyl chloride, which is responsible for liver and lung cancer. So the chemicals that cause cancer in these types of chemicals cause cancer in different manners, and the time between exposure and initiation of cancer can be large, so it can be varying the duration. It can be very difficult to identify the cause of the cancer sometimes, and the cancer causing agents are discovered from incidence of populations exposed to abnormally large concentrations, example industrial exposure, or from studies with laboratory animals. Okay, so this, it is a very difficult problem to identify the cause of cancer, and they are basically discovered from when the population is such that large population has been exposed to abnormally large concentration through basically industrial exposure, such as the Chernobyl disaster basically, for example, or from studies with laboratory animals, so then you can control them in a certain environment. So let us go to the next lecture, which is risk assessment methods. So basically, non-cancer effects. So the chemical effects of non-cancer, they can have an effect on target organs, which can be liver, nervous system, kidney, etc. Second is the reproductive system, and the third is development. So these are some of the targets, and how to basically quantify toxicity. So it is natural to quantify how toxic a chemical is, so we can control the levels of these pollutants entering the environment. So it is important also and natural also to quantify the toxicity of a chemical, so that we can control the levels as well as levels of these pollutants which is entering the environment. And the toxic effects is equal to potency, it is a function of both potency and exposure. So it is equal to potency into exposure. So potency is the vulnerability, and exposure is the duration. So it is not just how toxic a chemical is, it is also how much an organism is exposed to the chemical. So both vulnerability as well as the duration. Okay, so how much an organism is exposed, that is exposure, and also how toxic, that is potency, is a combination. Toxic effect is a combination of both. So how do you quantify toxicity? So when data regarding the toxic impact are gathered from laboratory studies or from epidemiological studies, information is gathered regarding the degree of toxic impact at different levels of exposure. So when the data regarding toxic impact are gathered from laboratory or various studies, information is gathered regarding the toxic impact degree, okay, at the various levels of exposure, so that we get a understanding of both. Okay? And this is the basically a graph which is showing that. So this is the dose and exposure relationship. So if you see here the response and exposure basically. So if we see here at the x-axis, basically it is no observable impact, okay, and after a certain time, so that is a threshold, and this threshold is called the NOAEL, that is no observed adverse effect level. So after that it starts increasing, and then uh, after it increases, the lowest point basically is the lowest observed adverse effect level or LOAEL. And after that it increases, and after finally it reaches a saturation, which is called the complete impact. So between complete impact and no observable impact lies the toxic impact, and the mid value basically, that is where 50% of population has been exposed to have certain effect. So that is EC50, or the location at which, or the exposure at which effect occurs in 50% of the population. So that is the general norm. Okay? But in the case of human epidemiological studies, so it may only have a few data points. Okay, so this is the ideal curve, but in case of human epidemiological studies, only a few data points may be available. So this is the actual real case of human epidemiological studies. So we can estimate the effect as a result of the exposure, but what exposure is acceptable? So that is a question. So we can predict the effect for us exposure, but what exposure is acceptable, we have to question. So this can be handled differently for cancer as well as non-cancer effects that also we studied earlier. And the non-cancer effects, in that case, the protective exposure is one with no effect. Okay, and the cancer effects, there the protective exposure is one at an acceptable risk. Example, one excess cancer in 1 million individuals. So the protective exposure at cancer effect is when uh, is one at an, which is an acceptable risk of 1 in 10 to the power 6, whereas non-cancer effect is protective exposure with no effect. Okay? So that is the difference basically. And the reference dose, as we studied, it is used for non-cancer risks. Okay, and the unit is basically milligram per kg day. And I also explained you this milligram is for the chemical or the substance which is toxic, and the kg is basically per unit body weight. Okay, so that is related to the body, and per day, so that is the unit. And it is typically a LOE, and as we studied, for converting from human to from animal to human values, we basically, the LOAEL or NOAEL, basically from the animal studies, they are divided by a series of uncertainty factors, often 10 cube. Okay, between 10 cube to 10 to the power 4, they apply a certain uncertainty factor, and the uncertainty factors account for unknowns such as difference in species, exposure duration, and also extrapolation, and many other factors are there. So basically within 10 cube and 10 to the power 4, we apply certain uncertainty factor. So, uh, for example, as per the Environmental Protection Agency, the website is shown here. So, for cadmium, the oral reference dose is 5 into 10 to the power minus 4 milligram per kg day water, and 1 into 10 to the power minus 3 milligram per kg day for food. Okay, that is the oral reference dose. And however, the inhalation reference dose has not been provided there. So the slope factor basically, that is used for cancer risks, and the typical unit is again fraction of population affected per milligram per kg day. Okay, so that is the percent of population of the total affected. So that is the typical unit, and uncertainty factors will be applied as needed. So if it is for the human animal data, we have to again apply uncertainty factor and convert it into uh, acceptable human value. So the extrapolation of slope factors, the question of how to extrapolate slope factors is often raised. So that is some of the interim understanding which is required for finding that. And much of the human carcinogenicity data is based on high exposures and high risk levels, and the data must somehow be extrapolated to lower exposures and lower risk levels. So based mainly the data which is available for human carcinogenicity, it is obviously for high exposure and high risk level. Then only people are more, that only people are more serious about. But it must be also extrapolated to lower exposure and lower risk level. So there also we need to apply some uncertainty factor probably. So this is an example. For example, it is a plot between excess cancer risks and exposure. So if we see here, so this is the linear extrapolation. So this is the ideal plot. But if you see the realistic plot, it can go both ways. It can curve out and also curve in. So in this case, if we see here, so the chemical is more potent at lower concentrations, okay, and then predicted by linear extrapolation. So even at lower exposure, it can has an it will have an excess cancer risk, okay. So the chemical is more potent at lower concentrations. Sorry, then predicted by linear extrapolation, so it can have excess cancer risks at a very low exposure also. However, in this case, the carving out, so if we see, the chemical is less potent at lower concentrations than predicted by linear extrapolation. So if you see here, even at a certain higher exposure, the excess cancer risk is almost it is less than 10 to the power minus 7, okay. So in here, even at a less exposure, the value is high. However, in this case, even at a certain high exposure, the value is less than minus 7, 10 to the power minus 7. So assessing the risk, so basically the toxicity information, that is RFD, reference dose, and SF slope factor for chemicals in a waste, along with estimates of exposure duration and frequency, can be used to assess the risk posed by a waste or contaminated soil, or can be used to set protective limits. So these are some of the things that which can be some of the requirements which can be kept in mind. So the information for chemicals along with estimates of exposure duration frequency can be used for assessing the risk, okay. So these are some of the data which can be used. And so these are some of the equations basically for calculating. So this is the equation for drinking water standard or target level, which is carcinogenic. So here the standard milligram per liter, so that is equal to TR into BW by CS F naught into WC, where TR is target cancer risk, having unitless, no units, we multiplied by BW, that is body weight in kg, and divided by CSF naught, oral cancer slope factor, which is milligram per kg day whole inverse, into WC, which is average water consumption rate, which is in liter per day. Similarly, the equation for drinking water standard or target level for non-carcinogenic substance, so that is C equal to RFD naught into BW by WC, where also C refers to standard milligram per liter. RFD naught is again oral reference dose, which is milligram per kg day. BW multiplied by BW, which is body weight in kg, and divided by WC, which is basically average water consumption rate in liter per day. So similarly, the example equation for clean soil standard or target level, where the substance is carcinogenic, and it is through ingestion route only, that is through mouth, okay. So in this case also, the standard C, that is in milligram per kg, and it is equal to TR into BW by CSF naught into IR, where TR basically refers to the target cancer risk, that is unitless. BW refers to the body weight, that is in kg. CSF naught is basically the oral cancer slope factor, that is in milligram per kg day whole inverse, and IR multiplied by IR, which is in ingestion rate, having units in milligram per day. So the risk assessment basically, so it includes both risk analysis and risk management. So the risk and analysis basically, so it includes 4 steps. That is firstly hazard identification. Second is dose response evaluation. Third is exposure assessment, and fourth is risk characterization. So these are the 4 steps in risk analysis, and that is followed by risk management. Okay. So firstly, four types of risk analysis, starting with hazard identification, then dose response evaluation, thirdly exposure assessment, and fourthly risk characterization, and it is followed by risk management. Risk assessment provides a qualitative or quantitative estimation of the likelihood of adverse unfavorable effects that may result from exposure to specific health hazards or from the absence of beneficial influences. So that is risk assessment. And management is basically, it follows the risk characterization as we just saw. It is followed by risk characterization, and it seeks to control exposures to toxic chemicals in the environment, and it also leads into law and policy for the same. So continuing the next lecture on risk assessment methods. So let us see some of the particular cases of some toxic substances. So how these are causing risks. Okay. So one is these are basically toxicants if we see. So one is polychlorinated biphenyls, which is PCB. Okay, PCBs. So these are most widespread chemical contaminant known, and they are discharged from factories into waterways. Vaporization from paints or landfills or burning of PCB containing material makes chemical airborne. It re-enters ecosystem with again precipitation. Okay, so vaporization from paints or landfills or burning of PCB containing material makes the chemical airborne. It makes the chemical airborne, and then again it re-enters the ecosystem with precipitation. And leaks from industrial equipment also is there, and also accidental spills and illegal dumping may be there due to high cost of illegally disposing of PCB waste, because it has a high cost related associated with disposing it in a legal way. So there might be accidental spills and leakage which is illegal, okay, due to illegal dumping. And the threat to health is laboratory testing shows toxicity even at low concentrations to several species that has been tested. And and that may result in chloracne, which is basically a skin disorder, and also endocrine disruptor. So endocrine is basically the glands secreting hormones into the blood, so it can also disrupt or stop the functioning of the endocrine. So this is an example of dredging operation, that is removal of soil from under the water or mud under the water. So dredging operation was conducted in May 2009 at Upper Hudson River in US to remove the PCB laden sediments from the river. Okay, Upper Hudson River. So this is an example of that. And one more toxic substance is the dioxin. Okay, so the full name is TCDD, that is 2378 tetrachlorodibenzo-dioxin. So it is shortly called as dioxin only. So it is formed as contaminant byproduct in production of herbicides. Okay, so such as example of that is Agent Orange, that was used to clear jungle vegetation during the Vietnam War, and that led to Parkinson's disease, ischemic heart disease, hairy cell leukemia. So these are some of the diseases, some of the problems, health related problems associated with dioxin. And also it can result in chloracne, skin disorder, variable toxicity. It has been found that variable toxicity was present in different taste species, and at high levels of exposure it is carcinogenic in most species, and also it resulted in endocrine disruptor and suppression of human immune system. So this is an example of the poisoning of dioxin in case of a former Ukrainian president. His name was Viktor Yushchenko. So his photo is shown here from. So if you see here, so in March 2002 he looked like this, and in December 2004 he looked like this. So that was because of the chloracne skin disorder coming from the dioxin poisoning. So he was scared by deliberate dioxin poisoning during his 2004 presidential candidacy. So this is so much harmful. Okay, so it has very ill consequences. So similarly, one more toxicant can be the asbestos. Okay, so basically this will not be toxicant because it is available in the this one. It is available naturally, so it will be a toxic substance basically. So it has probably caused more deaths and disabling disease than any other hazardous substance, and it is a group of six fibrous silicate minerals which is found almost worldwide. Okay, so it is mainly used in the construction industry. Okay, so this is used to reinforce clay and cloth. Since the stone age itself, and one of its properties is it is fireproof. Okay, so 85% of asbestos used today is incorporated into asbestos cement construction materials, and the problem is when it is inhaled. Okay, the fibers lodge in air passages and cells within lungs and blocks them. Okay, so that results in various types of problems. So if you see here, this is a photo of Libby, Montana. Okay, so it is a town of 3,000 people along the Kootenai River in USA. So it has emerged as the deadliest Superfund site in US history due to decades of contamination with asbestos fibers from a near vermiculite mine. So vermiculite is basically a micaceous substance. Okay, so those are used in the cement also. It has a very good water absorption property. Okay, so it can soak a lot of water. So there also the asbestos fibers are present. So so it was this place was highly contaminated, Libby, Montana. So because of a presence of large amount of asbestos fibers. Okay, and it has related into contamination due to the vermiculite mine basically. So the asbestos related diseases are various, such as scarring of lung tissue, okay, and lung cancer, leading cause of asbestos related mortality, and its exposure increases risks 7 times, with cigarette smoking risks is 60 times greater. So just having asbestos, it can increase risks of lung cancer by 7 times, and with combination of asbestos and cigarette smoking, the risk can increase up to 60 times. Okay, and also it can result in mesothelioma, which is a cancer of lung or stomach lining. Asbestos is also only known cause for that, and it is invariably fatal, generally within 2 years of diagnosis. So if anyone is diagnosed with mesothelioma, so the person can die within 2 years of that diagnosis. Okay, so it is such dangerous. Okay, such fatal. And here is an example of workers who in full body protective gear had dampened, that is tried to moisten the environment and remove asbestos containing materials from a high school. So such was the concentration of asbestos, such that they had to dampen the area so that it did not get inside their nose or mouth. Okay, so this is one of six substance, and one more is the lead. Okay, lead. So lead basically impairs brain function, and it results in hyperirritability, poor memory, sluggishness. Okay, so that is slowness, higher levels which cause mental retardation, and epileptic convulsions, coma, and death. Okay, so the house pain remains most notable source basically of lead poisoning problems. So the most of the lead poisoning problems is basically coming from the house paint. Okay, and as per statistics, 40% of lead ingested by preschoolers, that is children going to school and before that age, so it remains 40% of lead ingested, it remains in their bodies, and versus only 10% in adults. So it is very dangerous for children, and it interferes with blood cell formation, results in kidney damage, and any amount of exposure carries some degree of harm, which increases as level of exposure increases. So this is it results in kidney damage, blood cell formation interference, and with more exposure, it can cause a lot of harm. Okay? And this is a data basically graph histogram, which represents in the left axis, it shows the left vertical axis, shows the number of children tested. Okay, so it increased with from 1997 to 2006, and again decreased in 2007. That is the number of children tested. And in the left vertical axis, here it is shown confirmed EBLL, or elevated blood lead levels, as a percent of children tested. Okay, so fortunately it was from 1997 to 2007. If you can see here, the percent had decreased. Okay, so from 7% in 97, it had reached about 0% in 2007. Okay, so this is the blood lead level. Okay, elevated blood lead levels in children. Okay, uh, which was tested between the duration of 1997 to 2007 basically. So this is the data of US children having age of 1 to 5 with elevated blood lead levels between 1976 to 2004. So here also, fortunately, between 76 to 1980 percent of children having blood lead level, elevated blood lead level greater than equal to 10 microgram per deciliter. So that was the percentage was 88.2. Okay, so very high percentage, and that reduced to 8.6. So there was a drastic change, okay, drastic reduction from 1976 1980 to 8891, from 88.2% to 8.6%, and that also further reduced to 4.4% in 91-94, and also further reduction from 99 to 2004 to about 1.4%. So that is a positive news. And it has been found that all the lead also had a problem. Its deposition was also in the teeth. Okay, so that is dentine lead basically. So when the dentine lead was greater than 27 ppm, so that here the histograms are shown in red, and when the dentine lead, that is lead in the tooth, when it is less than 5.1 ppm, so it is shown in green. So it is seen that almost every behavior such as distraction, non-persistence, dependence, hyperactiveness, impulsiveness, frustration, daydreamness, does not follow simple directions, unable to follow sequences, low overall functioning. So every problem, every characteristic negative. So for the dentine lead having greater than 27 ppm, it has the maximum reported by the teacher. Okay, so maximum the negative traits were for those children which had dentine lead greater than 27 ppm, and lesser for those which had the dentine lead concentration less than 5 ppm. So it also had an adverse effect that way also, okay, in children. So proceeding with to the next lecture, which is on environmental risk assessment. So the risk assessment and bioavailability. So why it is important? The advantages are that if contaminants aren't bioavailable, then they aren't included in the evaluation of risk. Okay. The optimization of cleaning goals, it is required with a possible cost reduction and more effective use of available resource, that ensures basically more effective use of available resources, and it can achieve more accurate defensible cleanup goals while ensuring protection of receptors. Okay, so if so basically those only contaminants are basically assessed which are bioavailable, and otherwise so those contaminants are only selected for risks which are bioavailable, otherwise they are included in the risk evaluation. So that results in the optimization of cleanup goal, and also results in cost reduction, because we are very much specific. Okay, it has to be bioavailable, and also that results in more effective use of available resources and more defensible, like more targeted cleanup goals, and also protection of receptors. Okay, because we are more focused. However, it also results in certain challenges such as uncertain acceptance by regulators, stakeholders, public. Site-specific results difficult to compare across sites because it will vary. Okay? And potentially higher site characterization costs, because for each site we have to uh, separately perform that evaluation, and it may also take longer to gain full understanding of site conditions because it is very much dependent of the site. Okay? So, various processes to consider for this risk assessment during scoping, okay, or investigation. So there are various types of uh, various type of factors which has to be taken into account, ranging from chemical, physical, and biological. So in case of physical, there are factors like bed transport, deposition, resuspension, bioturbation, that is movement of soil, okay, advection, diffusion, grain size, COPC, which is cobalt halocyanin, distribution, burial temperature, etc. In chemical, there is sorption, that is absorption or adsorption, and desorption, transformation, degradation, geochemical, which is TOC, total organic carbon, salinity, pH, redox. And in case of biological, it can have uptake, transformation, bioaccumulation, mode of action, and critical body burden. So these are some of the factors which need to be taken into account for scoping or investigation for risk. Okay, and the sediments are basically mixtures, so there also we have to apply caution. Okay, so it is not a individual element. Okay, so here are some of the so this is a basically a figure of Anacostia River. So that is it is a place in Washington DC, USA. So it is a conceptual site model. So here we will find various factors. Okay, in terms of in in form of this locations. Okay, so various processes are happening here simultaneously. So this is the best example where you can take into account all those factors which are there, physical, chemical, and biological, for the investigation of risk basically in this river. The importance of quantitative measurements basically. So why quantitative measurements are important? So they basically help in defining the problem, and the reliable results from various lab and hands. And once the extent of problem is quantified, the fix can be designed, and they are the basis of research studies, and they are also used in finding answers to new problems and better answers to the old one. Okay, so these are some of the importance of quantitative measurements, okay, over qualitative. And the character of environmental problems, so relationships between living organisms and their environment. So these are some of the characteristics. And analytical procedures used are a strange mix of chemical and biochemical methods. This we will look into the this we will look about in the coming classes. Okay, and there is interpretation of data which is related to effect on microorganisms and or human being, and many of the determinants fall in realm of microanalysis. That is, we have to go for microstructural characterization because it is not possible to find the determinants by the naked eye. Okay, so here comes the requirement of standard methods. Okay, so standardization of methods for analysis is a requirement. So in the initial days, different methods were proposed for the same determinations, and that led to the difficulty in comparing the data which was coming out from various methods. Hence, the need of a comprehensive manual of standard methods, which everybody could refer to, was very much important, and that led to the development of standard methods of water analysis, which was uh, suggested by three organizations, that is APHA or American Public Health Association, AWWA, which was American Waterworks Association, and WEF, that is World Economic Forum. And the standard methods are basically accepted in courts. Okay, so when there is legal dispute, these methods will help in uh, providing a solution basically. So this is a chart. This is a basically a figure which shows the contaminant levels in receiving waters in the various decades. Okay, so if you see here, the in 1960s basically the majority contaminant level was basically due to bacteria, sediment, and BOD, that is biochemical oxygen demand, and the concentration was also very high. In the 1970s, basically the problem was with nutrients and CHLA, that is chlorophyll A. So the concentration started reducing. In the 1980s, the problem arose due to metals, persistent organic pollutants. Okay, so it was also further less. And in 2000s, the problem was related from the contaminants were mainly EDCs, which was endocrine disrupting chemicals, and PPCPs, which was pharmaceuticals and carbon care products. Okay, and so that EDCs and PPCPs were the main contaminant levels in 2000s, and from 2010 onwards, the new contaminant level was ENM, okay, which was engineered nanomaterial, and it had the lowest concentration. Okay, so with time, the concentration started decreasing, and also new types of contaminant had emerged. Okay. So with this, let us complete the lecture series for the second week and go to the solve problems. So the first question is, which of the following is correct with regards to the framework for sustainability analysis? A. Sustainability issues are complex and understanding can be established by well-structured analysis frameworks. B. Risk-based frameworks are emerging and gaining prominence for characterizing sustainability. C. Life cycle frameworks have been traditionally used to evaluate environmental issues and associated risks. Or D. All of the above. Okay, which is the correct option? A, B, C, or D? So if you see here, hint, the sustainability issues are complex, okay, and the understanding their interactions are equally important. With equally complex engineered systems is best done through well-structured analysis frameworks. Okay, so if you see here, the first option, it is correct. Okay, sustainability issues are complex and understanding can be established by well-structured analysis frameworks. Okay, the second, if you see, risk-based frameworks have traditionally been used. Okay, so it is traditional, not it is not emerging, okay, it is traditional, and it is used in characterization and prioritization of environmental issues, okay, not characterizing sustainability, okay. So risk based frameworks are traditional and gain prominence for characterizing your environmental issues, okay, characterizing and prioritizing environmental issues. And also so therefore the B option is wrong, okay. And the third is, increasingly however, life cycle frameworks are gaining prominence, okay, as a means of characterizing and understanding sustainability. So life cycle frameworks are not traditional, okay, so they are emerging, okay, so they are gaining prominence. And they are used to evaluate environmental, they are not used to evaluate environmental, but they are means of characterizing and understanding sustainability, okay, not environmental issues and associated risks. Or it is used for characterizing and understanding sustainability. So C option is also incorrect, okay, so the correct option is A. If you see, A is the correct option. That is, sustainability issues are complex and understanding can be established by well structured analysis frameworks. Second question, what is the meaning of risk assessment? Okay, first option, determination of the probability that an adverse effect will be produced. B, a methodological approach where chemical exposures are identified, analyzed for dose response relationships, and quantified for risk estimates. C, the process by which the form, dimension, and characteristics of risks are estimated. D, all of the above, A, B, C, D. So, which is the correct option? So if you see here, risk assessment is the process by which the form, dimension, and characteristics of risks are estimated. Okay, so that means the third option is correct. The process by which the form, dimension, and characteristics of risks are estimated. So C is correct. It is the determination of the probability that an adverse effect will be produced. So if you see, it is a determination of the probability that an adverse effect will be produced. So A is also correct. C and A are correct. And risk assessment can also be referred as a methodological approach where chemical exposures are identified, analyzed for dose response relationships, and quantified for risk estimates. So if you see B option, a methodological approach where chemical exposures are identified, analyzed for dose response relationships, and quantified for risk estimates. So this is also correct. Okay. So A, B, C all are correct. Therefore, D all of the above is the correct option. Okay. So D all of the above is the correct option. Third question, as per US EPA 2003, the reference dose RFD for fluoride in drinking water is 0.06 milligram per kg day. If the calculated average daily dose ADD is 0.086 milligram per kg day for fluoride with 3 milligram per liter concentration in drinking water consumed at a rate of 2 liter per day by an adult of 70 kg weight, which of the following is true for hazard index HI? A. HI is less than 1, acceptable. B. HI is less than 1, cleanup is required. C. HI is greater than 1, acceptable. Or D. HI is greater than 1, cleanup is required. A, B, C, or D, which is the correct option. So if we see here, if HI is less than 1, it is acceptable. Okay, so their cleanup is not required. So B is not correct. Okay, and if HI is greater than 1, then cleanup is required. We have studied that. So that is, it means it is not acceptable. Okay, so that means C is also not correct. Okay, so B and B and C are out of the box. Okay, so the correct option is between A and D. Okay, so B and C both are incorrect. So let us see which is correct. So if you see here the hazard index calculation, so for fluoride the hazard index HI will be ADD by RFD. ADD is average daily dose and RFD is reference dose. So average daily dose is 0.086 milligram per kg day. Okay, and reference dose is 2 into 3 by 70. Okay, so that comes about that comes to be 2 into 3 divided by 70. So it comes to be 0.06 milligram per kg day. Okay, so we are getting a value of 1.43. So that means HI is greater than 1. Okay, so when HI is greater than 1, cleanup is required as it is not an acceptable condition and possesses risk. Okay, so that means D. Okay, out of A and D is the correct option. HI is greater than 1, cleanup is required. So as you can see, D, HI greater than 1, cleanup is required is the correct option. Fourth question, what is the difference between toxicant and toxin? Okay, first, A, toxicants have acute response while toxins have chronic response. B, toxicants are released by living organism while toxins are anthropogenic. C, toxicants are anthropogenic while toxins are released by living organism. D, toxicants have higher latency than toxins. A, B, C, or D, which is the correct option? So if you remember, we discussed it properly. So toxicants are basically uh, substances which are released from man-made activities or anthropogenic activities, and toxins are basically those which are released from the living organisms. Okay? So that is C basically. Okay? C is the correct option. So if you see hint also, the toxicants are anthropogenic, produced by man-made processes, while toxins are released by living organism. For example, DDT is a toxicant, okay, dichlorodyphenyl trichloroethane, while snake venom is toxin. Okay, so toxicants and toxins both can be acute or chronic with varying latency. So these are not the correct options. Okay, so correct option is C. As you can see, C, toxicants are anthropogenic with while toxins are released by living organism is the correct option. Fifth question. Assume a student working in a laboratory accidentally exposed to a toxic chemical and immediately feels pain. The toxicity related to this accidental exposure can be called A. Acute toxicity, B. Chronic toxicity, C. Latency, D. Toxicity. A, B, C, D, which is the correct option. So if you see here, it says that a student who was working in a lab accidentally exposed to a toxic chemical and immediately felt faint. Okay, so the latency is very less. Okay, so because the toxicity, we are asked about the toxicity, type of toxicity, so the latency is not the correct option. And because type of toxicity is asked, so toxicity is also not a correct option. Okay, so C and D are incorrect, so it will be within A and B. Okay, so here if you see, because after getting exposed to the toxic chemical, the student got immediately fainted. Okay, so because the student got immediately fainted, therefore the toxicity exposure was short term. Okay, so that is acute. Okay, so therefore acute toxicity or A, acute toxicity is the correct option. If you see, the effect of the chemical is experienced immediately after having short latency time between exposure and response, hence it can be characterized as acute toxicity. So therefore A, acute toxicity is the correct option. So A, acute toxicity is the right answer. Okay, next question. choose Choose the correct order for the following process. A, risk 1, risk management, 2, exposure assessment, 3, dose response evaluation, 4, hazard identification, and 5th, risk characterization. So we have to choose which is the correct order for the following process. Okay, so first basically is the if you remember, first step is hazard identification. Okay, so that is number 4. Okay, so therefore A and D are eliminated, so it will be between B and C. So after hazard identification, we go for dose response evaluation. Okay, so that is 3. Okay, so if you see here, so between B and C, C is the correct option. Okay, so we go for dose response evaluation. After that, we go for exposure assessment. Okay, so that is 2. So 4, 3, 2. Thereafter, we go for risk characterization. That is 5. Okay, and lastly, we go for risk management. That is 1. Okay, so hazard identification, dose response evaluation, exposure assessment, risk characterization, and risk management. Okay, so 4, 3, 2, 5, 1. Okay, so C is the correct option. So here also it is written the risk analysis process includes 4 steps. 1, hazard identification, 2, dose response evaluation, 3, exposure assessment, 4, risk characterization, and 5thly, risk management is incorporated after above 4 steps. Okay, so basically C is the correct option. As you see, C, 4, 3, 2, 5, 1 is the correct order. Okay. Seventh question, match the contaminants with their source of release to environment. A1, lead, 2, asbestos, 3, PCBs, 4, dioxin, and the source of release to environment is A, uncontrolled burning of garbage, 2, B, house paints, gasoline, C, construction material, D, discharge, leak, or spill of industrial waste. Okay, so which is the correct option? A, 1A, 2C, 3B, 4D, B, 1B, 2D, 3C, 4A, C, 1A, 2B, 3D, 4C, or D, 1B, 2C, 3D, 4A. Okay, A, B, C, D, out of which is the correct option? So if you remember, lead is basically the main source of lead is house paint. Okay, house paints, gasoline. So one is basically B. So 1B. So A and C are incorrect. Out of B and D, one will be the correct option. So asbestos 2, it is basically it's coming from the construction material. Okay, so 2 is C. So if you see D, B is incorrect. So it will be 2C. So uh, asbestos is construction, that is D. Okay. Similarly, if we talk about PCB, it is coming from the discharge, leak, or spill of industrial waste. Okay. So polychlorinated biphenyls. So it is coming from the discharge, leak or spill of industrial waste. 3 is D. So as you can see here, and dioxin is coming from uncontrolled burning of garbage. Okay, so dioxin is basically coming from the uncontrolled burning of garbage. So basically 4 is basically A. Okay, so you can see all the options are matching to the sources. So 1B, 2C, 3D, 4A. D is the correct option. So here also it is shown. Correct contaminant source pair is given below. Lead, house paints, gasoline, asbestos, construction material, PCBs, discharge, leak or spill of industrial waste, dioxin, uncontrolled burning of garbage. Okay, so basically D is the correct option. So as you can see, D, 1 lead, house paints, gasoline, 2 asbestos, C construction material, 3 PCBs, polychlorinated biphenyls, D discharge, leak or spill of industrial waste, 4 dioxin, A uncontrolled burning of garbage. Okay, so that is the correct option. Eighth question: choose the correct statements about the characteristics of the toxicants. Okay, so choose the correct statements. A toxicant may or may not be bioavailable. B. Toxicant must cause metabolic dysfunction. C. Toxicant must not bioaccumulate. And D. Toxicant must interact at the molecular site. Okay, A, B, C, D. Which of them are the correct statements? So first statement is toxicant may or may not be bioavailable. So that is a wrong statement because it has to be, it must be bioavailable. If you remember, so A is the wrong option, incorrect option. Second, B. Toxicant must cause metabolic dysfunction. So that is a true. It is true. Toxicant, it must cause metabolic dysfunction. So B is correct. C, toxicant must not bioaccumulate. So that is wrong. It must bioaccumulate. Okay. So C is incorrect. Okay. And the last D, toxicant must interact at the molecular site. So that is true. It must interact at the molecular site. So basically B and D are the correct options. Okay. So as you can see here, the characteristics of toxicants, it must be in the environment, must be bioavailable and bioaccumulated, must interact at a molecular site, must cause metabolic dysfunction, and must decrease the fitness of an individual organism. So both are there. Must interact at a molecular site, must cause metabolic dysfunction, and it must be bioavailable. So A is incorrect, and also it must not bioaccumulate. So that is also incorrect. Okay. So both B and D are the correct statements. So as you can see, B, toxicant must cause metabolic dysfunction, and D, toxicant must interact at a molecular site are the correct options. Ninth question: which of the following biological processes are considered during scoping of toxicants? A, oxidation, B, bioturbation, C, bio transformation, or D, bioaccumulation. Okay, so which of the following biological processes are considered during scoping of toxicants? Okay, so we are basically asked about the biological processes. Okay, so here if you see the bioaccumulation, biotransformation, so these are the biological processes, okay, which needs to be considered during scoping of the toxicants. Other than that, the bioturbation, that is a physical phenomenon, so it is not considered into biological process, and oxidation is a chemical process, so that is also not considered into biological process. So biological processes are basically biotransformation and bioaccumulation. So C and D are the correct options. So as you can see, C, biotransformation, and D, bioaccumulation. So these are the correct options. And last question, which of the following are incorrect when RISK assessment is done based on bioavailability of contaminants? Okay, so which of the following are incorrect when RISK assessment is done based on bioavailability of contaminants? A, only when the contaminant is bioavailable, they are considered in evaluation of risk. B, the cleanup cost is high when only bioavailable contaminants are considered for cleanup. C, it does not achieve more accurate defensible cleanup targets. Or D, site characterization cost is high and it takes long time to gain full understanding of the site. Okay, so out of A, B, C, and D, which is the which are the incorrect risk assessment related statements based on bioavailability of contaminants? So if you see here, only when the contaminant is bioavailable, they are considered in evaluation of risk. Okay, so that is true. Okay, so that is not incorrect. So A is not the correct option. The cleanup cost is high when only bioavailable contaminants are considered for cleanup. So that is wrong. Okay, cleanup cost is low. It is cheap when only bioavailable contaminants are considered. So B is the correct. B is one of the correct options. C, it does not achieve more accurate defensible cleanup targets. So that is wrong. It achieves more accurate defensible cleanup targets because it is specific. So C is also incorrect. So that means C is a correct. B and C are the incorrect statements basically. And D, site characterization cost is high and it takes long time to gain full understanding of the site. So that is a true. Okay, so A and D are true statements. So that's why they are not the correct statements. And B and C are false. Okay, so they are basically the incorrect statements. So as per the question, we have to see the incorrect statements. So B and C are the correct answers. So here also the hint is given. When the risk assessment is done based on bioavailability of contaminants, if contaminants aren't bioavailable, then they aren't included in the evaluation of risks. So he is correct. This leads to optimization of cleanup goals with a possible cost reduction. So this is wrong. On other advantages include more effective use of available resources, more accurate defensible cleanup goals. Okay, so this is wrong. While ensuring protection of receptors. Challenges to this approach include higher site characterization costs, so this is true, and longer time to gain full understanding of site conditions, so this is true. Alongside site-specific results are difficult to compare across sites. This approach sometimes leads to uncertain acceptance by regulators, stakeholders, and the public. Okay, so B and C are the incorrect options. So these are the correct options for the question. Okay, so as you can see, B, the cleanup cost is high when only bioavailable contaminants are considered for cleanup, and C, it does not achieve more accurate defensible cleanup targets. So these two options are the incorrect and or the correct answers for the question. Okay. So with this, we come to the end of today's class. So let us meet again next Sunday with the third week lecture notes and problem solving discussions. Okay. So with this, I would end today's discussion. Thank you.